This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 70 for February 19th, 2010. Hello, everyone. I'm Vincent Racaniello, and it's time to talk about viruses at the top of the food chain. Back again after a year-long absence in Argentina, just a month. It seems like a year. Is Dickson Despommier.
It did seem like a long time.
Welcome back, Dick.
Thank you very much, Vince. It's a pleasure to be back.
Professor Emeritus.
That's right.
Here at the School of Public Health.
And
Columbia
Department
University.
of Microbiology.
How does it feel to be emeritus?
The whole month of January when I was on vacation, and for the first time in my life, I felt relieved.
And let's bring in Alan Dove in Western Massachusetts. Welcome, Alan.
Good to be back.
So that you can rib Dickson as well.
Go for it.
So
guys.
I hear emeritus leads to hiatus. Is that word?
That's right.
What did you just tell us what you did in Argentina?
We actually wandered the countryside seeking interesting opportunities for visualizing the wildlife, which included condors, flamingos, guanacos. We looked for fishing opportunities, of course, which we found many of. We tried to get out of the wind, which was steady at about 50 miles an hour with gusts up to 80.
What's that called? There's a name for that wind, right?
I call it Mariah. They call the wind Mariah. I call it... You don't want to hear what I call it because fly fishing people have bad names for the wind because it's not your friend. And there were times where I was standing in the middle of a river. The wind was blowing upstream so hard it was making whitecaps on a river. And my rod was bent also upstream, and I could not tell whether I had a fish or whether it was just the wind.
Alan,
I had never experienced
Don't say anything, Alan.
Nope. I'm...
Gentlemen, you obviously have to do this to appreciate it. And then we saw the most awesome amount of scenery that you can possibly imagine.
It's a beautiful country.
We saw the Fitzroys, which are a series of mountains named after...
The Fitzroys.
Who was he?
Gerald Fitzroy.
What was he?
I don't know what he was.
He was the captain of the Beagle.
No.
Yeah.
I didn't know that.
Yeah. So Darwin's presence in that area is widely known.
That's cool.
And we also saw the Torres del Paine, and this is a series of magma chambers which hardened underground and then by an uplift was pushed straight up through the surface of the earth. And they stand like stacked plates. And it's amazing. These big, sort of an oval of rock that goes up for thousands and thousands of feet, and you can see snow on them. And it was just the most impressive scenery. We were within about 100 miles of the Straits of Magellan. Pretty
far down, right?
I've never been that far down in my life.
So you wandered all over.
We did. We rented a car. We took some back roads sometimes, and we saw wildlife virtually everywhere. We saw so many condors. We got, oh gosh, there's another condor. And we saw tons of flamingos, which I had never expected to see, actually. And
Were they pink?
They were, with black wing. The back edges of their wings, the flight wing feathers were all black. They were so beautiful. You could see them standing out in the middle of these wetlands, and the wind was so fierce that they had their heads tucked up underneath their wings, and they were braced into the wind. And to see them actually like that, and then, of course, the nice way to get to see them is to scare them. So they all start to fly, and then you get to see what they really look like. It was quite amazing, actually.
Did you take pictures?
Lots.
Well, you'll have to back them up onto your Drobo, Dick.
I shall.
You don't want to lose them.
No, we won't lose them. They're all over the place right now. So, and then, of course, we caught some amazing trout, so it's a fantastic place to go down for trout fishing as well.
Yeah, we have a picture of you with a trout, right?
You do. Should
we put that on the website?
Damn right. Let David Baltimore eat his heart out.
Yeah, he's a religious listener of TWiV, sure.
I'm sure he is. If you are, David, we didn't mean it.
Yeah, we don't mean it. Well, we have some wonderful stories today, Dick.
I'm anxious to hear them.
And let's get started.
Yep.
The first one, Dick, you'll love this, as so will Alan, published in PNAS. A broad-spectrum antiviral targeting entry of enveloped viruses.
Huh.
Have you ever heard of a broad-spectrum antiviral?
No.
Do you know that there are broad-spectrum antibiotics for bacteria, right?
Sure.
They hit gram negatives and gram positives.
Yep.
But our antivirals are pretty specific. But this is a very interesting new one that came out of a group at UCLA mainly, the head author, Ben-Hur Lee, and many other authors. What they were doing is they were looking for an inhibitor of Nipah virus entry. So they screened a library of chemicals, and they found one, which they call LJ001.
Yes.
And this is an aryl methyldiene rhodonine derivative.
Sounds like a dye.
It does sound like a dye, doesn't it? And it turns out, this is what I'm showing Dick what it looks like here.
Oh, my.
A couple of rings. Not too
Heterocyclics.
complicated.
A lot of heterocyclics.
They found that it inhibits all enveloped viruses that they checked.
It's amazing.
They add it to the virus, and then they
I
infect
see.
cells.
I see.
It inhibits.
It blocks the receptor.
No, it doesn't block attachment. It blocks some post-attachment step, possibly
Post-attachment,
fusion.
I see.
It blocks. Listen to this cast of viruses that they tested: Ebola and Marburg,
Wow.
influenza A,
Ooh.
arenas, Rift Valley fever and lacrosse, which are bunyias, flaviviruses. Dick, what's your favorite flavy?
I guess I'll have to say West Nile.
It inhibits West Nile, Hep C, yellow fever,
My
paramyxos,
God.
Nipah, and parainfluenzas, retroviruses, HIV-1, rhabdos, and pox viruses, but no effect on viruses that don't have a membrane,
Huh.
like adenovirus, Coxsackie, and reovirus. Isn't that cool?
It's very
That
cool.
is really cool.
I think this is terrific.
You have to mix it with the virus first, though.
You do have to mix it with the virus.
That does present a little problem.
I would say it does.
So what they think is happening is that it inserts into the viral membrane and damages it.
Which isn't actually the viral membrane, is it? It's
It came from the cell, right?
Right.
And that's probably why it
a
hits all of
modified
these.
host.
Yeah, and that's probably why it hits all these different viruses, because the thing they share is an envelope from the cell. Right?
Exactly.
And it probably damages it sufficiently that the
Yep.
virus can't fuse.
By the way, there's a... Mind if I interject here, just because I'm back for the first
You
time?
can always interject.
Thank you. The malaria parasite, when it exits from a red cell,
Right.
is surrounded by portions of the red cell membrane.
Is that right?
It's,
Do
an,
they-
env-
is
it's
that-
an enveloped protozoan.
And does it remain with the parasite?
Yeah, until it enters the next red cell, and then it sheds that
Oh,
one and
cool.
it picks up another one.
That's interesting. Stealing from the viral world.
Well, it's a theme that's worth
Hmm.
repeating.
Anyway, so this compound... All right, so you have to add it to virus, but if you put it in the medium, it will block the virus from spreading from cell to cell because the viruses that are produced from
Got
the
it.
first cell are inactivated. But this is different from other antivirals or many others. It doesn't get into the cell and block replication. It actually binds the virion.
Right.
And it binds to cells, but it's not toxic.
Let me ask you a question here, then.
Did you hear that?
I did.
It's not toxic.
That's
I did.
very cool.
What's the
I heard
question,
it.
Dick?
The question is, then, could this be used as a way of using live virus vaccines?
How so?
Well, you have a live virus and you're afraid to give it to an animal because you don't want to start an infection. You don't have to do that. You can mix it with this material first and then give it to the animal.
Inactivated. It depends how
And now
stable
you've got a
that
live
is, yeah.
virus that gets in and stimulates immunity, and you don't have to worry about inactivating the virus.
But if this is effective as an antiviral, then you wouldn't get enough replication to stimulate immunity.
Or it might be dose dependent.
Yeah, you'd have to
Might be.
put more virus in. Which,
Just a wild thought.
I mean,
Yeah.
it's not a bad idea, but that's the point is, yeah, you wouldn't get enough replication.
Right.
But if you give a big enough bolus,
That's the point.
maybe that would
Yep.
work.
Yep.
Many questions to be addressed here.
Interesting
Why it
stuff.
doesn't affect cells is very interesting.
Yeah.
So their theory is that the cell membrane, even if you damage it, it can fix itself.
Sure.
It has regenerative properties.
You bet.
They call it bioregenerative.
Maybe that should be the definition of life.
Something that can bioregenerate.
Yep.
Yeah, because the viral membrane cannot regenerate.
That's right.
Yeah.
Hmm.
Hey, never thought of that.
So
Well, that's a
is
good
cool.
one.
Never thought of that until just now.
Something that can repair itself.
Exactly.
Okay.
Sustainable.
Cars can't repair themselves.
Well,
Ergo, they're not living.
Puncture-resistant tires can.
They're living. We got living tires on our cars.
That's right.
Now, so this is cool, and this will go forward. They'll do toxicity.
Absolutely.
They'll do
Right.
derivatives, pharmacokinetics.
Of course.
Right. And the fact that this compound exists tells you that there's probably a whole
More.
realm of chemical space that you could
Sure.
explore, modify.
Absolutely.
if you do see toxicity in animals, you can start tweaking it and see what you can do with it.
Wow.
Now here's...
So wait a minute, I have a question
sure.
it's still part of this article.
Of course.
How would you go about doing a computer search for a compound that would aid you in this quest to block the virus replication? Well, I mean, what was the information that they fed into the computer, and they said, well, when it comes back this, then we know we can test this compound? What were they looking for?
Well, they had a library of compounds, so they had a collection.
But what were they basing the library on?
That's a good question. We can probably find it in the supplemental materials.
Something that would integrate into the membrane of the...
Well, that's a good... I'm not sure that they did. Let's see. That's a good question, because
by the way,
there are different kinds of libraries.
So it's no wonder that this is a broad spectrum. It's actually not a broad spectrum because it's the same membrane in all those viruses.
I understand. Yeah.
Okay, so they started out with a high-throughput screen for inhibitors of Nipah virus.
Yeah. Did they say what kind of chemical library they used?
That is probably going to be buried deep in materials and methods.
Or deeper.
Or deeper, yes.
Or deeper.
Data not shown.
Data not shown, that's right. See next paper.
Yeah, I'm looking at the supplemental data, and they don't tell the origin.
How would you organize the research? You're thinking about, Jesus, I wish I had an inhibitor of Nipah virus.
Yeah,
great.
Well, no, there's a whole industry built around this. A whole sub-industry of the pharmaceutical industry that does high-throughput library synthesis and screening.
Yeah. But so that sounds random. This one doesn't sound so random.
Well, you can have molecules that target membranes in libraries.
Yeah, okay, and high-throughput screening has gotten a lot less random in recent years. They've the initial efforts at it were really just sort of shotgun approaches, it's easy to synthesize. But now they've gotten a lot smarter about exploring the chemical space and trying to define structural moieties and classify the types of compounds they're looking at. So the libraries tend to be a little more tailored. But I'm not seeing any commercial affiliations on here. So I don't know where the library came from.
Yeah, they don't say. So it could have been for membrane active, but maybe not. We don't know. We're guessing.
Well, if Ben-Hur or any of his colleagues are listening, and by the way, cool name.
Not bad. It's a classic.
Ben-Hur Lee is the senior author.
Aha.
Just send us an email. Let us know where the library.
Yeah, we'd like to know the thinking behind the result.
Yeah.
The lead author is Mike Wolf.
Yeah.
And then there are many in between, and I assume that's because they tested many viruses.
Right.
Here, okay, my final question.
Huh.
Do you think viruses can evolve to be resistant to this antiviral?
Because it's not a viral component, it's a host component.
Well, there are examples of antivirals that target host components, and you still can get resistance.
This sounds like a general exiting strategy for viruses, though, of the enveloped type. So how could you possibly know that that is the Trojan horse that you're carrying with you to the next cell?
Well, my knee-jerk response is if you have a compound that kills a virus, the virus is capable of eluding.
One would suspect that the answer
just
is yes. That's right.
But the question is, what is it going to cost the virus to do that?
Yeah, it's a good point. I think you're right. I think that it will evolve in some way, but it may be sick.
Yeah.
They do a limited study in this paper where they pass HIV a number of times in the presence of this compound and don't see any resistance, but it's a very limited experiment.
Right.
I think that if it's not a matter of whether resistance, but when.
Always the case, I think. We've never been proven wrong on that one.
And what the impact is. Because if the virus can adapt to this, but then it can't really mount a fully successful infection, then you're still okay.
Sure.
Yes.
It might slow it down way below the horizon of clinical medicine.
Right.
Yes. The cost.
And that will undoubtedly vary from virus to virus.
It will.
Exactly. Just the implementation is the problematic part for me. I can't see how this would be used in clinical medicine.
Well, you would, I don't know if it's orally bioavailable, but you could take
That's
it
going to be
orally.
a big hurdle.
It may not be.
You might want to give it during an outbreak to the people that haven't been exposed yet or have the potential for being exposed. But
It could be.
I don't think it would be a general...
The thing that might be useful is that you don't need to particularly diagnose the specific infection. If it hits all envelope viruses, you may be able to treat quite broadly. And if you don't get resistance, then you can use it indiscriminately.
And it could make sense to dose people after they've started to show symptoms, because if you are inhibiting subsequent rounds of replication,
Gotcha.
then you're going to shorten the course. I mean, that's essentially what you're doing with Tamiflu.
That's right.
So have they tried that out in clinical trials in animals then?
Well, this is brand new stuff, so they
Right,
first need to do some toxicology and pharmacokinetics in animals and decide. There's a lot of work to be
there's
done.
tons of work.
But it may be a derivative of this that eventually is used. This is a lead, right?
And this may not go anywhere. In fact, the overwhelming majority of leads don't go anywhere.
Correct.
That's right.
But this is a very interesting lead.
Have to follow it.
I think it's a good proof of concept that you can have a broad spectrum inhibitor.
Right.
And in biological terms, how it works is going to be very interesting to sort out. Because it may lead to other molecules once you know the mechanism. Right?
Yeah, that's right.
They say, by the way, that when you add the compound to the virions, they remain intact, so it doesn't just disrupt them. Just kind of interesting.
Yeah.
There's also, there could be some good basic research uses for this, too.
Sure.
I mean, you can do, if you give a large enough dose of it to your cell culture, you can do a single round of infection.
That's right.
Which could be very useful. You could get a single synchronized round, and you know that everything happened in that round.
Yes.
Right.
Exactly.
So if you want to study entry or what have you.
Yeah, even if it fails as a drug, it may be useful for research, and that's happened before, that's for sure.
Yeah,
absolutely.
The Win compounds that inhibit picornaviruses are useless clinically, but Alan Dove used them for his...
I used them in my thesis, which
a big win.
Which I guess was not useless.
Not at all. Absolutely.
Still.
We published papers, and you are
That's right.
You are a learned writer as a result.
That's right.
That's right. Virologist in residence in Western Massachusetts.
That's right.
Okay, I think we will move on to our next stories, but before we do, we have a couple more interesting ones. I want to acknowledge the support of TWiV by Data Robotics Incorporated, the makers of the Drobo, which is a wonderful storage and backup solution for your computer. They make units of different sizes, which are basically racks for hard drives, but they're beautiful. They're black and shiny, and you can put 4 or 5 or 8 hard drives in them of your choice. Plug them into your computer.
I have a Drobo.
Dick has a Drobo now, and we will put your Argentine photos on it, so they will be protected against any catastrophe except fire.
So protection,
if your house burns
Drobo,
down.
You should call it Drobo Cup.
Drobo Cop, oh my gosh.
Sorry.
Yes, Dick,
Sorry.
we will. I will help you with the Drobo.
Thank you.
If you wish.
I wish.
You can put how many photographs would you say you have? Over 50,000?
I don't think I'm up to that, but
I thought I saw 25 to 30,000.
I have a lot of them.
We will put them all on the Drobo, and they'll be safe. Because if any drive fails, this is how the Drobo works.
Yes.
You just pull out that drive, and you know because on the front a red light comes on. You pull it out, and you pop a new one in, and all the data are restored. Because everything is stored redundantly over the whole array of 4 or 5 or 8 drives. It's brilliant technology.
Sounds great.
Artists use it. Photographers are well known to use Drobos. Scientists, not so much. So all the scientists listening, go on over to drobo.com/TWiV and check it out, because we just got one for the lab here. I'm putting all my data on it, and I know it's going to be safe.
Yep.
And it's in one place, not on CDs all over the place, for example.
Exactly.
It's great for labs that generate a lot of data. For example, labs doing systems biology experiments. I understand that a single experiment can generate a terabyte of data.
Wow.
So a Drobo will someday, well, the 4-bay Drobo, when they make 4 terabyte drives, that'll store up to 16 terabytes of data. So right now the 8 drive model will hold 8 terabytes, or 8 times 1.5 is 12. That's the biggest. Actually, there's a 2 terabyte drive available now, so that could hold 16. So you can do your systems biology experiments. And
Fabulous.
If you make movies like Avatar, you generate terabytes of data a day, and you
right.
can store them on these as well.
Exactly.
Anyway, so Drobo is a proud sponsor of This Week in Virology. We very much appreciate their support, and what they have done for us is to give us 3 units to give away to listeners of TWiV.
Yay!
So you should head on over to twiv.tv/Drobo contest to find out the rules. We're giving one away each month, so in February we're working on the 1st, and the rules for that contest are at the website link I just told you. Basically, you need to subscribe to TWiV in iTunes and write a review in iTunes to be, and also do a tweet, which will then allow us to find you, and then we're going to randomly pick one. And the next 2 months, we'll devise some other contest, like a 300-word essay on the meaning of viruses. No. We'll let Alan read them all.
No.
No.
Both of them.
Alan would want a Drobo just to read them.
That's right.
Yeah.
Anyway, that's drobo.com/TWiV. We thank them for their support of virology, and specifically this week in virology. Now, the second story we have, I think you found this, Alan. Is that correct?
I think that's right, yes.
Cucumber mosaic virus hacks aphid behavior to maximize its spread. Ooh. Do you want to handle this, Alan?
I think I can, yeah.
Excellent. There you go.
So these folks, let me get their affiliation. They're at Penn State, and they're entomologists.
Great school. Great school.
Yeah, very good school.
Yep.
So they were looking at feeding behavior of aphids, and they determined that aphids, if you release aphids onto a plant that's been infected with cucumber mosaic virus, CMV, not to be confused with the CMV that humans get.
Right.
So you infect the plant with this virus, and you let aphids feed on it, and the aphids feeding on those plants will promptly leave those plants and fly to plants that are not infected, if they have the option. So aphids don't like to feed on the plants that have this virus. It's apparently a lower quality, less attractive plant.
We don't know why, right?
We don't know why. This is just the observed behavior of the aphids.
Okay.
So if you force them to feed on these plants, they are not as healthy. The aphids are not as healthy.
Mm-hmm.
So apparently they're getting less nutrient value from them. And I guess they have ways of sensing this, and they bolt and head off to plants that are not infected. Interestingly, if you let the aphids choose, and they haven't fed yet, you give them the option of choosing plants that are infected and plants that are not infected, they will go first to the plants that are infected. So those
even
plants,
though they're nutritionally less.
even though those are worse plants to be on, and even though once they arrive and start feeding, they want to
going
leave.
for the Big Mac rather than the healthy salad.
That's right. That's right. So it looks good from a distance, and then you get it, and you say, that's really not quite what I wanted.
There's so much like that in life, isn't there, Alan?
Yes, they're really.
Come on, we have to lighten this up a little bit. This is a serious topic.
No problem.
All right. Well, I'm trying to fit a Tiger Woods joke in there.
No, he's come clean, so I think that
true.
your days of making fun are over.
Okay, so the apparently the plants smell good to the aphids, and the aphids go to them. And then once they start feeding on them, they figure out the plant isn't so good after all, the infected plant, and then they go on to an uninfected plant. Now, what these folks did was they actually analyzed the volatile compounds being released by these plants.
It's amazing.
After they saw this behavioral phenomenon, they said, this is kind of interesting. So looking at the volatile compounds that are being released, you find that the infected plants release more of the volatile cues that would attract these bugs. But even though these plants are actually less attractive, the plants smell better.
Right.
And this has an obvious evolutionary advantage for the virus. So
how so, Alan?
Okay, so the virus infects the plant, causes it to release these compounds that are going to attract more of the vector. I guess that's a piece of the story that I didn't provide yet. These aphids are vectors
for the virus, right?
So the virus, on infecting the plant, it comes in on an infected aphid, infects the plant, and immediately causes the plant, or soon after infecting it, causes the plant to produce compounds to attract more aphids.
Amazing.
But at the same time, it makes the plant less tasty to those aphids. So once they arrive and start feeding,
they go sooner.
And they pick up the virus, then they want to go find an uninfected plant.
It's all connected.
They bring the virus
exactly
with them.
What benefits the virus. They take the virus with them to an uninfected plant, start feeding, and
amazing.
This is the way we view it, right? But there could be some other rationale for this. But in our view of the situation, it looks like this has evolved to spread the virus, to transmit it.
This is called viropocentric thinking.
Exactly.
Yes.
I mean, they say at the end of this article that we need to study more examples to make sure this is what's going on.
And that there's not an advantage in doing this for the plants.
Right.
Or the aphids.
Yeah. So if you could manipulate the system in some way to alter this, would be good. If you could alter the production of the volatile compounds.
I think the first thing you need to do is see if you can alter the virus.
So it doesn't induce these compounds.
So it doesn't induce these compounds. See what its spread is in a test plot.
Yep.
And if you actually are reducing the spread of the virus, then that would be strong evidence that this is an evolutionary trait of the virus.
Sure.
So Dick, it says here, vector-borne parasites can induce changes in the traits of their primary hosts that affect the frequency and nature of interactions between hosts and vectors. So this is an example of that.
Yes.
Do you know any in the parasite world?
Well, I just read 2 of them right here in the article itself. I was unaware of the fact that Leishmania-infected or Plasmodium-infected people smell different to their vectors than non-infected people.
Leishmania-infected hamsters
attract
sandflies because they smell different. And Plasmodium is with people. Plasmodium attracted significantly more mosquitoes than uninfected children. How would a parasite induce volatiles?
I don't know, but that might be a red herring because with Plasmodium falciparum infection, you're a little bit slower, therefore less likely to move away from a situation that you're being bothered by mosquitoes in, for instance.
Which is the one where the mouse behaves differently and the cat eats?
Toxoplasmosis.
So the toxo in the mouse makes it behave differently?
Yes, it eliminates its fear of cats.
It stays there and they kiss and eat, and then he eats them.
Right. Unless they're Tom and Jerry. There's some remarkable relationships out there, and they're all related to these things. The one I liked the best appeared on the cover of the American Scientist one year. It showed a tobacco plant being eaten by a tobacco hornworm caterpillar, which evolves into a moth afterwards, right? And it also showed an ichneumid wasp coming into the scene, and the ichneumid wasp is really a parasitic wasp that was attracted to the damaged plant because the plant gives off a pheromone-like molecule which attracts in the parasitic wasp. And once the wasp is within view of the tobacco hornworm caterpillar, it finds a target for its eggs and implants them inside the body of the caterpillar, and eventually the larvae end up killing the caterpillar.
Yeah.
So it's like the plant calls in the police that come in and arrest the criminal for stealing their, it's amazing stuff.
Do we know any examples in human virus infections of this sort of altering the behavior of the infected host to facilitate transmission? I don't know of any.
I can't think of anything.
No.
Studied in parasites and plant viruses, for example, but I'll bet there's something in people that...
Another relation... You're asking me to think outside the box here because these are not viral examples, but there must be some equivalent to that in a viral infection. The other one that I like really well is a tapeworm that cats acquire by eating frogs. And cats, they love to eat frogs. If you throw a frog in front of a cat, it'll pounce on it, except the frog will jump away, of course. So there's this one particular tapeworm that as a larva lies under the skin of frogs and secretes growth hormone-like molecule, which allows the frog to increase of its size to the point where it slows down.
Can't jump away.
And the cat catches that frog every single time.
Terrific. That's terrific.
It's called Spirometra mancinoides. That's the name of this parasite. But sure, there must be tons of infections that are related like this. Otherwise, they wouldn't be spread from animal to animal. There has to be a mechanism other than the environment to account for the high rate of infection for some of these things.
Well, it may be when someone gets influenza, people feel sorry for them, so they stay around to help them. And
Ebola works like that. I mean, people, they had a cultural habit of actually kissing their dead relatives before they buried them. And everybody associated with the outbreak was dying.
Well, listeners, if you know of any examples of this behavior modification in infected people, let us know.
Right.
Well, I can think of certainly biological effects in infected people with diarrheal viruses. Any gastrointestinal virus that causes diarrhea is going to increase the spread of that virus in areas with poorly treated sewage.
But in terms of interaction of hosts with vectors, I don't know of any, but there must be some people who think about this.
Absolutely right.
Okay. That's very neat. I love that. Yep. I like the title you gave it. cucumber mosaic virus hacks aphid behavior to maximize its spread. Hacking aphid behavior. That could be a title for this
There you go.
show.
That's right.
We're always looking for catchy titles.
Absolutely right.
One guy wrote me once, he said, "Do you name these this way to get people to listen?" I said, no, but maybe it catches your eye and then you will.
Well, look at all the names for genes that they've got. Come on, you've got hedgehog and hawks and all these names that people use as euphemistic ways of referring to the very complicated.
Sonic hedgehog.
Sonic hedgehog. That's all based on cartoons,
Yeah. Well, life is a cartoon, isn't it?
Life is a cartoon.
Our next story is another Alan find, new vaccine technology.
Wow.
Yeah.
This is so
Alan,
cool.
you've been busy.
I get these press releases all the time, and every now and then there's something good in them, so I
This
have
is
to
so
keep
cool.
reading them.
This is just beautiful for you
This-
guys who like
this, I think,
engineering.
is-
Talk
Yeah,
to us. Talk
I
to
think
us.
this is absolutely awesome. So, one of the big problems in delivering vaccines, especially to poor countries in the tropics, is that you've got to keep them refrigerated.
That's right. A
Most-
cold trail. You have to have a
Yeah,
cold
exactly.
trail.
You got to have the cold chain intact.
That's right.
This is especially true with live virus vaccines because
Yeah.
the virus has to arrive
Measles
alive.
is like that, isn't it? Yeah.
Right.
Measles, polio, sure.
Yeah.
Measles, mumps, rubella, the whole thing is infectious.
Interesting.
Polio.
Right. So you've got to stabilize these things by keeping them cold.
Right.
And what these folks did, and this is Adrian Hill's group at Oxford, and we have talked about some of their work before, I think. So they came up with this method. They observed that sugars are used often as stabilizing agents in biological products.
Right.
Like when you're freezing something down, you can use
Sure.
sugars as stabilizing agents.
Yep.
So they took sucrose and trehalose and made sugar glass from them. And they did this. They mixed these sugars in with virus preps, and they chose an adenovirus and a pox virus that are used as vaccine vectors for new generation
Very
vaccines that are being
interesting.
developed. So they mixed the sugars together with the viruses, slowly dried them down, and made this sugar glass, this crystallized sugar that has the viruses crystallized in it.
Which must taste good, too.
It probably does, but they dried it down on a membrane,
Oh, okay.
plastic membrane, and found that they could leave that sitting at 45 degrees Celsius,
Wow.
which is hot,
Yep.
for 6 months, and they could still recover full viral titers. from it.
Full. No loss of infectivity.
No
By
loss of
just
infectivity.
dissolving it?
Yeah, then they just put
Yeah,
water
they
in
just-
it.
they
dissolves.
rehydrated it, threw it on cells, and it still had full titers.
Whoa.
Here's the glass here, a picture of it.
Mamma
Yeah,
mia.
so then they got together with Nova Biopharma. It's a company, and they made this neat little syringe device. It goes on, I guess, with a lure lock fitting just on a standard syringe. So you've got the membrane with the virus dried down in this sugar, and you have the syringe attached.
Oh, look at this.
Then you could just suck the saline. You could deliver this thing, throw it in shipping crates, and send it across the ocean and send
Right.
it to wherever, Somalia.
Yeah.
And then presumably you just draw some sterile saline up into it.
It mobilizes the
It
virus.
rehydrates.
Back
and
out
it
again.
delivers somebody's vaccine.
That's outstanding.
See that little that little membrane is what the sugar film is.
That's outstanding, Alan.
Yeah.
That's great
So they
stuff.
say this doesn't making this sugar film doesn't impose all the drying that you would normally do to freeze dry a vaccine, for example, which
Right.
imposes chemical stresses on it. This is very gentle, and you get rid of all the water, which is what you want to do.
Sure.
I mean, the temperatures they use 45 and 55 degrees centigrade.
Yeah, they didn't take it easy on this virus either when they were testing it.
It's very hot.
Be darned.
So of course now the thing to do is to take currently used live virus vaccines.
Yeah.
Yeah, take a polio vaccine, and
Yeah.
the polio vaccines which
marvelous.
you drink, you could put it on these and just pass some saline through it and drink it.
Yeah.
Right, and then now you eliminate the kerosene fired freezers that WHO had
Sure.
to develop.
Or you can make a children's candy out of it.
This is actually sugar.
Yep.
It's
Yeah.
fabulous. This is a great melding of technology.
Love it.
Yeah.
Because the virologist wouldn't think of this. They have to talk. So this fellow has dabbled with the powdered measles vaccine before. And
Right.
We talked about that once on TWiV.
Hmm.
And this is beautiful melding of virology and engineering.
Right.
I love this. And the numbers are very convincing.
Yeah.
And I like this statement here at the end. This tells you that they're Brits, okay? The temperatures and time periods used in the study do not accurately mimic the unpredictable fluctuations that might be encountered by a product during shipment, which could happen, for example, in the hold of an aircraft or the boot of a car.
The boot.
The boot.
Which is the trunk, right?
Yeah.
Yes.
So they say we have to investigate the stability under normal temperature fluctuations that might occur.
Right.
But this is very promising.
Yeah, so they just have to give it the boot.
Wow.
Give it the boot. That's right. Boot it up.
Or if you're in Canada, you have to be out and a boot.
It's in a boot. Yep. Very nice paper. I
Love
like
it.
This now let me rant a little here.
Rant.
This comes out in a journal called, what is it? Science Translational Medicine. It's a new journal of sciences, right?
Yes.
We don't subscribe to it here at Columbia.
Oh.
Why is it so hard for me to see this paper?
Where did you have to go to find it?
I had to email him a PDF.
Alan Dove. I asked Alan Dove, who's got access.
Okay.
It just bugged me that I couldn't get access. And it's for a good purpose, and it's behind a wall.
I see.
There has to be a way around this. I know Alan has to get paid.
We have restrictions that didn't allow us to actually subscribe to this one.
Well, they didn't subscribe to it. They don't, the university has a limited budget, so it doesn't subscribe to every new journal that comes out.
Wait a minute.
We don't even subscribe to Cell, Host, and Mike.
Did you say we had a limited budget?
It's not infinite.
That's not how we spend here.
Yeah, I know. I mean, there are people out there in the world that would like to read this and they can't. I think it's a shame that research like this, which has broad implications, can't be read by everyone. Not everyone can understand it, of course, but I wish we could work around this.
Yeah.
I don't know what the solution is. We've talked about that before here, but
well,
anyway,
you see,
Alan
Google
was
is about to go publish popular books online now.
Yeah.
That's going to cause some authors to get bent out of shape.
Eventually, there will be a solution. It takes time.
Mm.
All right, and then I think we could quickly deal with this last story. Do you want to do that one, Alan, before we move on to email?
Sure. So this is a study that just came out in retrovirology, and this is some folks at the MRC in Great Britain looking at the XMRV CFS link. And we've talked about this on the show before.
Yes,
XMRV is a recently discovered virus, and a little while ago, one group found that infections with this virus I guess infections with this virus correlated with cases of chronic fatigue syndrome.
Exactly.
And then another group found that it didn't. And somebody found that XMRV correlated with prostate cancer, and then another group found that it didn't. Right. And now we have another group finding that XMRV does not correlate with chronic fatigue.
Right.
So
caffeine causes cancer. Caffeine cures cancer. Caffeine.
Yes. So now one of the issues that came up with the first study with the chronic fatigue and XMRV was that this organization, the Whittemore Institute, was on the original paper that found this link, and immediately, as soon as the paper went to press, the Whittemore Institute was offering tests for XMRV, and right. they're a chronic fatigue treatment facility, which now I don't like to go on conflict of interest witch hunts, but I have to say it's a little unseemly when you're selling a product that stems directly from research that hasn't been replicated yet.
Correct.
So they, of course, say that the British groups are not doing their PCR reactions correctly. The British groups, of course, have a different version of events. And I think that at this point it's time for an exchange of hostages. What you need to do is take somebody in the British side, send them over to the Whittemore Institute, and take somebody, send them to the other lab, and have them duplicate each other's techniques and see where the problem lies.
By the way, there's an example of this in nature. That's the Judah Volkmann laboratory up at Harvard had this problem with proving certain anti-angiogenic factors were created. No one could replicate it, but every time they went to his lab and they did the experiments, they replicated it. So it turned out to be an environmental issue.
Yep.
Very, very interesting.
These things happen. I mean,
they do.
But this came up actually in the early days of molecular biology. I think there's a theory about maybe it was Hershey was doing some of the original
Yeah.
reps, and nobody could duplicate his techniques, and then they came to his lab and they found out that he was using a much lower grade of ether than everyone else, and
Yeah,
that
that's right.
the contaminants were actually important. So
Yeah.
Well, in this case, the Whittemore is claiming that the Brits don't know how to do PCR properly.
Right.
Which is, to me, I don't know. It's PCR. It seems to me quite straightforward, but really exchanging samples would help.
Yeah.
What will not solve this is for the groups to accuse each other of not knowing what they're doing.
Correct.
Right.
That is not how you resolve problems in science.
That's right.
You resolve them by doing experiments,
Yes.
right?
This is true.
Please, let's all do this in a reasonable way.
That's right.
I mean, the problem is that the Whitmore is highly invested, right?
Yes.
And they want this to be true, and I think that's unseemly, as you said.
Yeah.
But let's do experiments, okay?
Yeah.
And go
Science
forward.
will out.
So this is an open question. I think it could still go either way.
It's true.
There's no definitive answer, but we just need more results, and I'm not involved in this.
Thank God.
I do not have a dog.
I.
I do not have a dog in this fight either. No.
And I'm rejuvenated, so I'm not tired at all now.
I mean, I hope it would be nice if this virus were a main participant, because then one could design therapies.
Exactly right.
Oh, sure.
And because I know there are a lot of patients who would like to see that.
Always better to have a diagnosis than not.
We don't know yet.
It's true.
All right, let's do some email.
Hey.
Have you missed TWiV email, Dick?
Have I? Have I?
You have to admit they are fun to read.
I love them.
Okay, Andre writes, "Hi, Vincent, Dick, and Alan." Well, he got the right trio for today.
That's right.
I recently listened to a Nature podcast interview of science journalist Simon Singh. He wrote an article about some very questionable claims of chiropraxis to cure diseases like colics in children, and found no evidence to support those claims. Apparently, he got sued by the British Chiropractic Association for libel. However, that brought him into trouble, and he finds himself now defending his position with great financial burden. The British law system is in that respect very expensive and problematic, and intimidates many authors to publish articles which might upset powerful communities like the BCA. Another weird thing is that even international authors can be sued for libel. And there's a petition on the internet which he links to. I am aware that your platform has no political interest and only deals with science, but I thought this case might be of interest to you as scientists, in particular science writers, like it is to me. I don't ask you to put this issue into your podcast if you don't like it, but if you want to touch on it anyway, I would love for you to discuss the freedom of speech for scientists on a general basis in situations where scientists find themselves arguing, maybe in court, with groups of totally unscientific, often crazy beliefs like creationists or people like that. Many thanks, and keep up the good work. What do you think, John?
Well,
should we have freedom of scientific speech?
Absolutely.
Based on your results, you betcha.
Yeah. I think what has happened to Singh is unconscionable, and the British Chiropractic Association should be ashamed of itself. I mean, if they actually had a legitimate objection, they could present the evidence for it.
Correct.
The courts are not the right venue for discussing this sort of thing.
No.
What do you think will happen in that case, Alan?
I think I'll get sued.
Come and get me. Come and get me. You'll never find me up here.
Well, like the BCA has a vested interest in this, right?
Yeah. I mean, obviously they want to believe that their claims are correct, and Singh claims that they're not. But this, to me, strikes at. Obviously the British system is different, but here in the United States, where I'm fortunate to be sitting, this is not the way things are done. We can say what we want, and is it libel? I haven't followed the whole thing, so I haven't read exactly what Singh wrote. But if he's made claims and backed them with evidence, then I would think truth is a defense.
You bet.
Well, here in the U.S., we have freedom of speech, period, for all areas, not just science, right?
Right, but we do also have libel laws. I mean, you can't come out and say something that's outright false and defaming.
Right.
Yes,
right.
That's true.
So in terms of science, if you have the evidence, the results to back up what you're saying, it's fine. If you have evidence that vaccines don't work, you can say that.
Right.
But you notice that in the U.S., there are many people who trash vaccines, but they don't get sued by the vaccine manufacturers.
Right. Because the vaccine manufacturers, unlike the British Chiropractic Association, understand that this is going to backfire.
Right.
Right.
And
Right.
if anybody from the chiropractic business is listening, I would say to you, this is a really stupid tactic. This is, regardless of what you think of what this guy said, suing him is not going to silence him. It's actually going to do quite the opposite. In fact, I wasn't even aware of this article. I know of Simon Singh. I read one of his books, but I didn't even know he'd written this article until I found out of the lawsuit. So.
Yeah, another a listener has recently emailed this to us as well. So I thought we should talk about it because I feel, as you all do, I'm sure that when you have the data, you should speak it out. Right? And if we find a drug, there's data that says that drug is not effective or unsafe, you release it, you publish it, you have it peer reviewed. Right. Now, in this situation, he did his own investigation, right? So he's not publishing, although Nature supported it, I suppose, right? It's a Nature podcast interview. He doesn't work for them. He's an independent journalist, right?
Right.
So I don't know. It's a little different as opposed to science freedom of speech because we're talking about, getting data and publishing them. What do you do with investigative journalism, Alan? How does that differ?
It would follow kind of similar rules that you have to back up your claims. You have to have your sources, and you have to be willing to cite those sources and say, this is how I'm justifying what I'm saying here.
Sure.
And again, I have not followed this issue closely enough to get into the nitty gritty of exactly what Singh said. But I would expect as an experienced science journalist that he knows the libel laws and that he would have, backed up his claims sufficiently to defend against them. And I just kind of a little point of order here. My understanding was that in the British system, if you sue somebody and lose, you have to pay their legal expenses.
That's right.
So I would think that this wouldn't actually be particularly expensive for him if he wins.
Right.
But I could be wrong.
Well, we're not lawyers here.
And no, we're not. This is not This Week in Law. So
that's right.
There is a podcast of that name.
Okay.
Anyway, that's good. Thanks, Andre, for pointing that out. And let us know, listeners, what you think. Pamela writes, sorry I didn't put this in the previous email, but I was just finishing TWiV 47, where Dick talks about torches. Still catching up after the birth of my daughter. Just a quick correction. Torch, T-O, toxoplasmosis.
Yes.
R, rubella.
Yes.
C, cytomegalovirus.
Yes.
H, herpes.
Yeah.
And S, syphilis. Dick listed something for the O.
Orphan.
Which I don't believe stands for anything. And he had listed hepatitis for the H. So H is herpes.
Well, I'm just quoting a physician that actually taught that to me, so I...
Well, she's a physician also.
I deeply apologize. Maybe these are older ways of spelling it.
She's a physician at NYU. Torches are infectious, a.k.a. preventable and or treatable causes of birth defects, which is why they are drilled into our heads in medical school and why every pregnant woman should be tested for them.
Hear, hear.
Someone else may have corrected this. If so, my apologies. No one else. Thank you, Pamela, very much.
Yes.
And thanks for listening.
Yep, I sit corrected.
Tom, the beekeeper from California, writes, I just finished listening to Original Antigenic Sin. That was last year, where you're talking about Wolbachia's protective effect against virus infection in Drosophila. I was delighted that you were speculating about the effect in bees because Wolbachia has been one of my favorite subjects for years. He actually has a wonderful email, which we should put on the website, but let's extract a bit of it. Beekeepers have used tetracycline routinely since the 1950s. Against a bacterial disease of the brood called American foul brood. A great name. This usage has almost certainly cleared any Wolbachia infections in domestic bees. So if they once had it, interestingly, we have now developed bees that are resistant to that disease, which make antibiotics unnecessary, though they are still used. Wolbachia has been found to be very common in African bees, but only in Africa. Okay, that's cool.
Everywhere else, too.
Another well-studied Wolbachia effect is something called cytoplasmic incompatibility. This can result in sperm produced by infected males killing eggs of uninfected females. Infected females, on the other hand, produce viable offspring using either infected or uninfected sperm. This gives infected females a huge reproductive advantage. Wolbachia is very selfish, and since it is passed on only maternally in the cytoplasm of the egg, it uses the genetically dead end male to kill off the competition. Wow, we're dead ends.
Wow.
Always have been. Either a dead head or a dead end.
My own pet hypothesis is that this could explain how descendants from 26 African honeybee queens released in Brazil in 1956 were able to make a nearly complete genetic sweep all the way to the southern U.S. Supporting evidence is still lacking since Wolbachia has yet to be found in this hemisphere, but they're looking. Could be useful for protection from viruses. And then he notes that a person we are working with to find Wolbachia in bees is David Schneider at Stanford, and he quotes an article of his from Science last year, which we'll put up. I find you and Dick brilliant in the way you come up with great ideas on the fly. It's a pleasure to listen to such intelligent people bounce ideas around. Thanks for such interesting program.
Well, let's repeat that email again and again.
Sometimes we come up with things on the fly, as well as our other
That was not a pun, I hope.
No. Dan writes, Vincent, well, thanks for a wonderful pod class. Very good experience. Wow. That's a nice one.
Yeah.
I'm a meteorologist in Colorado, and my daughter is in grad school, microbiology with a biochem undergraduate degree, and might get into virology. Time will tell. She is just starting her first lab rotation. I thank you for your podcast, as I can now converse with my daughter more on her science level.
Great.
I have a broad education in the physical science, but zilch in biology until your podcast, and she is the other way around. But now I can talk some bioscience with her and kind of make some sense thanks to you folks. Now to the point of this email. So he thinks he had H1N1 in October by symptoms. It wasn't diagnosed. The quick test at the physician's office didn't work. And so he went home not knowing if he had flu, but he thought he did. And he was given an antibiotic, which I knew not to take. And yes, our doctors still need help in clinical infectious disease. That is a topic for another day. So then he got the vaccine a couple weeks later, H1N1. And he said he's always had a reaction to flu vaccines, swelling, soreness, but he had none to this. So he says, is that because I was immune to H1N1? And then the vaccine was eliminated without a reaction? Or is it coincidence? He said, none of the people I talk to can give me a clear answer. I'm sorry that we won't be able to give you a clear answer either. Because
I can give you an unclear answer if you'd like.
An N of
I'm good
one.
at that, apparently.
It could have been that you had immunity. 3 weeks, it's kind of borderline whether you'd have sufficient immunity to, dampen the effect of the vaccine. On the other hand, if you have a good immune response, you might get a reaction as well to the vaccine. Because that's what happens when you have a vaccine and you have a reaction. It's usually your immune system kicking in.
Yeah.
So this could have been made more severe by having previous immunity. On the other hand, you could have been infected with another virus, and the H1N1 vaccine perhaps didn't give you a reaction this year because it didn't have the contaminant that you're reacting to. So, sorry, Dan, there are just too many variables. That's the way science is.
Or after many years of flu shots, you've finally gotten tolerized to the defense of them.
Or they forgot to put the virus in the vaccine that time.
So is it correct to say, gentlemen, that the science works when you look, you set up experiments and you test them. You don't look at anecdotal data and make conclusions.
That's right.
The only thing you can do with anecdotal information is say, gee, that might be worth investigating.
That's right.
The best experiments are the ones that you don't have to interpret.
Yeah.
Yes.
Thanks again for the good topics. I now listen weekly in my car commute and burn your MP3 files to CD for my daughter to listen to at some point in the future when she has more time. She should listen to them now. Because that'll really, if she's a micro grad student, she should listen to them.
And in the future, we're not going to have CDs anyway.
Nope.
There you go.
Won't be able to read them. P.S. You mentioned in one episode that you use ensemble models in your field. We use them now in meteorology to better judge the probabilities of a modeled forecast. Maybe we will have more common scientific ground in addition to dengue fever outbreaks related to weather patterns in the future. Dick is going crazy. Thanks for your diligent production of these programs, and know that I will always be listening. Hey, cool. That's great. Thanks, Dan.
Absolutely.
At least we'll always have one listener.
At least.
Sam writes, hi, Dr. Despommier. Do you know Sam?
I don't know.
He's here. Look at his name.
Sam looking at his name. Wait a minute. I have to get my glasses.
All right,
for this
Robert Desowitz has some new books, at least ones you didn't reference on TWiV.
Probably right.
His book, The Thorn in the Starfish, was a great review of the immune system with historical anecdotes.
Uh-huh.
You would love Federal Body Snatchers and the New Guinea virus. It's right up your alley and perfect material for TWiV.
Have to read
it. It opens with the emergence of West Nile virus in the U.S.
Now I have to read it.
And very similar to your first TWiV,
Yeah.
and concludes with an insightful description of the U.S. patent law system.
Interesting.
Many great stories in between, including the tale of the CDC patenting antibodies that naturally occur in New Guinea people.
Wow.
Unfortunately, I can't find my copy of Federal Body Snatchers, so I can't be specific in the details, but I think a discussion on biotech and patenting human genes antibodies would make an excellent TWiV topic.
Yep.
Yeah, that's a good idea. I love that you're starting a new podcast that includes eukaryotes. It would be good if you discussed this classic on TWiP. And it's this paper, Trichuris suis therapy for ulcerative colitis.
Yeah.
You know about this?
I do.
We'll talk about it on TWiP.
Yeah, well, he's one of the authors, I see.
No, he's not an author.
Yes, he is.
Where? That's DE, and this is Sam.
Well, maybe he's using Sam as a
All right. Okay,
nom
Dick,
de plume.
I am applying to master's degree programs at Epi schools in public health. I've applied to the University of Michigan's molecular Epi program, Tulane's parasitology program. Do you have any suggestions? My interests are tropical diseases and eukaryotic parasites.
Right.
Ideally, I would like to work with someone researching the molecular epidemiology of a parasitic disease.
Hopkins. Johns Hopkins is a great school.
Yeah.
And UCLA has a great school. Yep.
Parasitology, epiparasitology. Yep. All right.
A lot of good people there.
There you go.
And by the way, his best book, which everyone quotes, is New Guinea Tapeworms and Jewish Grandmothers.
Right.
And let me just tell you an anecdote about Bob Desowitz. Bob and I are good friends, and at one of the national meetings we were at, which was in Puerto Rico, which was a fabulous scene to have a national meeting at, Bob and I decided to play tennis one afternoon, and he's a fantastic tennis player, and I hadn't known that, of course, and he's older than I am, and I thought I'll whip this guy's ass in no time. He beat the living god. He beat
stuffing. He beat the stuffing out of you.
He beat the heck out of me. A parasitologist can use euphemistic terms like that, but I wouldn't dare do it on the air.
All right.
But he's a wonderful guy and a great scuba diver and a wonderful observer of science life.
Excellent. All right, let's do a couple of emails from our students in Indiana University of Pennsylvania. I don't know if you were here for this, but a virology class was given an assignment, Dick, to listen to TWiV and send us questions.
Really?
So all the students have been sending us questions. So here's some more. We've read a few. Stephen writes, TWiV, I am an undergraduate at Indiana University of Pennsylvania. In the questions section of TWiV 66, you say that it would be impossible to remove the endogenous retroviral DNA in the human genome.
Right.
If it were possible, do you think there would be any phenotypic changes in a human without these elements? If so, what would the changes be?
Excellent.
They would be 3 inches taller, with longer hair and blue eyes.
Right.
Guys, so these are kids trying to learn stuff.
Well,
we don't know.
We haven't got it.
Some of us believe that there would be no changes because it's all junk DNA.
I don't believe in anything that nature keeps around and replicates that is useless. It's just we haven't found the function for it yet.
I think if you removed, I think some of them probably are things that we wouldn't miss. I suspect that there is actually some genuine junk DNA because, unlike Dick, I do think nature keeps some things around that we don't actually need.
Why?
Evolution is bureaucratic. It adds things very easily, but if there's not much downside to keeping it around, it's not really going to go away.
You know what? We should have a debate on this one because I think the situation warrants the fact that situation where it's necessary isn't always present, so you keep it for the emergency.
But in this case, we're dealing with retroviruses that got into the genome not in our interest, and then lost the ability to replicate after propagating through the genome.
Yeah.
So they got there because they could, and they haven't been deleted because they didn't need to be. So they may not ever have provided anything useful.
How do we know way back, I mean,
we don't.
Way back, that these were spacer molecules or something else that allowed the, transcription factors to fit better or something, I keep
But then how do you
things.
explain something like the fugu genome? which has almost no junk DNA.
I'm not going to explain the fugu. Just don't eat 2 pieces of it, that's all. Or is it 3? I forget which.
Maybe they were never infected with a retrovirus.
Right. But it shows that complex life is possible without.
Sure, that's a good point.
Sure.
So there are genomes without endogenous.
And look what you get. You get a poisonous fish as the result. So when they remove our...
Oh, but they're cute.
The interesting thing here is that the koala has been recently infected with a retrovirus within the past 100 years.
Yes.
And it is going into their germline. So the koala existed without endogenous retroviral sequences, and now they're only going in probably because they were infected. And whether it has any effect on them, we won't know for millions of years, of course. But it could be that they're of no use. I don't know the answer, Dick. I can see both sides of the argument.
But the other thing is, if they're in there, it's likely that DNA has then been co-opted for something.
Maybe.
I mean, at some point. So I would suspect that if you could somehow remove all of them, if you could, create this minimal human genome, I would think it probably would affect something.
Well, almost half the genome is these mobile genetic elements related to retroviruses.
Right. If nothing else, it would affect chromatin condensation.
Yeah. So you couldn't take them all out, but if you could take a few out, you might be interesting. And you could do that in the mouse, which has endogenous sequences, and you could take them out, but I don't know that anyone's done that.
Right.
But it is a very interesting, thought-provoking question.
Are any of them repeated again and again and again, like 10 repeats or multiple repeats of themselves?
I don't know. They jump around because occasionally you make active reverse transcriptase, and then these elements can move, and they duplicate that way, and that's why they're called mobile genetic elements. And they do cause diseases, and some people think that the antigens made are involved in various diseases.
So wouldn't it be interesting when all of us get our genome sequenced?
Yes.
To compare our non-coding regions to see if it's all the same.
I don't know if you get the non-coding sequence when you do that. Don't you just focus on the coding region?
Well,
I mean, that's the non-redundant, non-repetitive stuff, because that's the easiest to assemble. Do you know anything about that, Alan?
I think when you do a whole genome sequence, you do a whole genome sequence.
You get it all.
You don't just do this.
Because how would you know until you finish?
Yeah, because I think a lot of the SNPs are in... that a few years ago there was this big hunt for single nucleotide polymorphisms,
right?
And I think a lot of those are in the junk DNA.
So did Watson's DNA compare favorably with Venter's DNA in terms of their repeated units that are considered junk DNA? Are they the same?
I don't know. They're not the same. No,
they're not the same.
There are SNPs that distinguish them.
They're
right.
not the same.
If they were identical twins, they would be very similar, though, right?
Well, that would be great to study them at birth and then see if they start to differ as they acquire.
They acquire some changes, sure. That would be interesting, but
or even over time in the same individual.
I don't think you would see much change in the lifetime of an individual, though.
Okay.
Katie writes, do you think it's possible for chronic wasting disease of deer to make the leap from deer to humans? Cervid wasting.
Yeah.
What does CWD stand for, guys?
Chronic wasting disease.
Chronic wasting of cervids.
Yeah. Of cervids.
It's a prion disease.
Yep.
Yeah. It only takes one amino acid change to change other species of prions. Right. So sure, it could happen. And I even go as far to say that it will.
Don't eat that elk.
Yeah, you shouldn't.
I don't know if I would say that it will, but I wouldn't bet against it.
Right.
I just wanted to provoke discussion.
I wonder if it'll get into the wolves.
Yeah, you can't say it will. We cannot be certain of the future. It's difficult to predict the future.
I always said it's difficult to predict the past.
But it could, sure. It's absolutely possible. And the more people eat infected deer, the more likely it becomes. And if the deer isn't terribly sick and you kill it and you don't know that it has a prion in it, then you're increasing the chances. I like her quote here at the end of her email. If at first the idea is not absurd, then there is no hope for it. You know who said that, Dick?
I would guess Einstein, but probably I don't know.
You would be right.
There you go.
You're brilliant. Elisha writes, hello, I am new to the study of virology, so my question may seem basic, but it is something I don't understand. One can be infected with a cancer-causing virus and yet never develop cancer. How does that work? What triggers some people to develop cancer and others who are infected with the same virus not to get it?
The answer is a Nobel Prize winning answer. Whoever figures that one out is the genius of the year.
What did you say, Alan?
I said bad luck.
bad luck, a change in environment, which allows it to happen.
I mean, our immune system gets rid of many infections with these viruses, right? Some luck is involved. Innate immunity, adaptive immunity, cellular.
Right.
In fact, our immune system gets rid of many nascent cancers.
Maybe all of them in healthy people.
Yes. So that's a big factor, our immune system.
You bet.
There are a handful of human cancer viruses, and that's probably something in common to all of them, that our immune system really helps.
Well, there are some triggers, though, right? Environmental triggers, like, for instance, with lung cancer and smoking, for instance.
And I don't know what virus causes
No, say it's not a virus. It's a genetic...
Well, that's different. But this is virus-induced cancer. So we have hepatitis B, hepatitis C, human papillomavirus, Burkitt's lymphoma by EBV, and HTLV1, right?
Right, but look at the difference between EBV and... senior moment here, folks. Just give me a moment. I'll come over. And mononucleosis. Exactly right. Thank you very much.
Yeah, well,
in Africa, it's this, and everywhere else, it's that.
It's mainly in Africa.
I know.
Right. Burkitt's lymphoma was
it's in Africa. It's in Africa.
Right. So there are environmental influences, population genetics.
It's got to be. And human genetics, the genetics of the individual that's got it.
Yeah. So does that make it clearer?
Multiple factors.
I think it's as clear as it's going to get.
Right.
Okay, and our last virology class question for today. We have more. Good afternoon. In podcast number 67, you discussed chronic wasting disease in deer caused by prions. I've read that all prion diseases, or transmissible spongiform encephalopathies that affect the brain or nervous tissue, are untreatable and fatal. Correct. The infectious form of prion, PRPSC, is able to infect and propagate by forming an amyloid fold. Have researchers been able to isolate the amino acid sequence or other factors that leads to the misfolding? Since non-infectious prion proteins are found in all individuals, is it possible to manipulate the amino acid sequence of an infectious prion to convert it to a PRPC?
Wow.
It's a good question. It is the same amino acid sequence, right? In both proteins, in the folded and properly, in the improperly folded protein.
Right.
The question is, I think one amino acid change causes the misfolding, right?
There was also...
Well, there's spontaneous misfolding, right?
Yeah, there's spontaneous misfolding, and there's been some discussion, I haven't followed it closely, but about glycosylation affecting the folding. So it could be same sequence, differentially glycosylated.
Yes. So this is how it happens. So you can have an amino acid change, which makes your prion misfold, and those have been identified in the genetic disease. In certain people, they have inherited or familial. But then there's an infectious form. You're infected with a misfolded prion, and that propagates the misfolding to your normal prions. How that happens is under study. We don't understand. So we know what kind of changes will cause misfolding, but we don't know how a misfolded protein propagates the change to a
It's a mystery disease. It definitely is a
People have been able to do that in vitro, I believe. You can take a misfolded protein and add it to a properly folded one and get it to misfold.
But I think you have to add some semi-crude cell extract to that.
Mm-hmm. Really? That may be so.
It's still not entirely clear what all the factors are.
That's a very interesting area of study. Look at Prusner's work, and there are many others. Just search prion in PubMed. Don't do it in Google. Search PubMed.
Yes.
And let's move to our science picks of the week. You must have accumulated so many in Argentina, thinking every Friday I could be picking them.
I actually thought of one as the aphid article was being presented.
What's that, Dick?
And it's a book called Chemical Evolution, and it's written by Dr. Eisner, and I'm blocking on his first name. I think it's Charles, but I'm not sure.
The guy from Cornell?
Yeah, the guy from Cornell.
Thomas Eisner.
Thomas. I have his book. And
he's the ant guy, right?
He's a lot of
entomologist.
Guys. No, that's E.O. Wilson. He's not an ant, but I think he studies chemical communication in insects.
And insects, okay.
And you're right. Ants are communicating by chemicals too. But the book itself covers a wide range of. That's the guy. He's a fantastic explainer of science.
Insects are our beasts of choice.
Wait, which book is this?
It's called Chemical Evolution. It's part of the Scientific American series of publications.
One of his popular book, For Love of Insects, was one of my picks of the week a while
ago. I have that book.
Yeah, great book.
He's got another book It may not show up because it was published by
Oh,
Scientific
chemical
American.
ecology.
Chemical ecology. I take it all back.
The
Chemical
chemistry-
ecology. Okay,
Chemistry
ecology.
of biotic interaction.
Take it all back. Take it all back.
What's
Chemical
that about?
evolution.
What's that about?
It's about the way nature communicates with itself.
Cool. That has to do with our aphids, doesn't it?
Aphids and bees and trees and plants and all kinds of things.
Excellent.
And us. And there's a lot of interesting examples. I wonder if he's aware of the fact that certain infections can give off, the infected individual can give off a different odor and therefore favor the transmission of that.
Mm-hmm.
You would be fascinated with that result.
Well, you can get a copy on Amazon for $367.
Or you can read it for free online.
That's right.
Is it available?
It is. It's National Academies Press,
Okay.
and everything that they publish is free in one form or another. So the deal is you can get the paperback version. You can order the paperback version from them.
Right.
The National Academies are government organizations.
They are, that's right.
You know,
They still charge for the originals, though.
well, you can, right, because it costs them some money to
But of
print.
course, of
So
course.
for $41.18,
Yeah.
you could buy it from them, or for $34, you can get a downloadable PDF.
Right.
Or for zip, you can go to the link that I just put into the agenda. We could put it in the show notes, and you can read the thing online.
Right.
All right. Hi, everybody. It's Vincent and Dickson, and we're back. And we had a little glitch
We had a happening.
recording TWiV 70.
That's right.
I had forgotten to plug in the laptop, so
Whoops.
ran out of power while we were discussing, I think it was your pick of the week, Dick,
Oh.
chemical ecology.
Okay, we can do that now because we've corrected all of the misinformation that I had about it to begin with.
Well, we talked about your pick, which is fine. And
Right.
then Alan, unfortunately, his pick was lost, but we could tell everyone what it was.
Sure, it
right?
was a book on mass spectroscopy.
It's a website called the Scripps Center for Mass Spectrometry.
Right.
And this is a website by Gary Shudak, who tells us all about what mass spec is. So
Yep.
why is mass spec important, Dick?
I think because you get sequencing data instantly from complex molecules.
Yeah, you can identify complex molecules in complex mixtures.
Yeah,
right?
you don't have to purify them.
And so Alan was saying that children at birth now are tested for
Fetal
diseases
ketonuria,
by
such
this.
as that. That's right.
It's used forensically. It's used in research labs.
Absolutely. It's used to sequence proteins.
So it's an important technique, and here's a good website to teach us about it.
Indeed.
Right? So that was Alan's pick, and I'm sorry we lost his audio.
Right.
My pick was inspired by a reader email from Jesper from Sweden, who said, and the pick is called Folding at Home. Dear Twivers, having followed and recommended your show for quite some time, I figured I'd give you a weekly pick that might be of interest to you. I do not recall you ever mentioning this one, so apologies if you know about it. I ran into something called Folding at Home, a technological relative to SETI at Home. Do you know what SETI?
I do. It's the search for extraterrestrial intelligence.
Intelligence, right.
I actually met the person in charge of it.
Really?
Yeah, they gave a talk at the TED lectures that I was at, and she received a special award for it. We had a long, long talk about it. It's very, very interesting stuff, actually.
Yeah, they use people's computers.
Yeah.
They put a little program on them and run them. Distributed computing.
What they're doing is analyzing radio signals from outer
Yeah.
space to see whether or not there's a perturbation that might indicate some pattern to somebody's message.
Folding at Home uses the CPU
We are
cycles.
coming to eat you.
It uses the CPU cycles not on radio signals, but on folding proteins.
Fabulous.
For the advancement of medical
Because
science.
it takes a lot of computing power, right?
It does. It's operated by the chemistry department at Stanford. And
It's a
he sent
fabulous
us
idea.
a link, and we'll put that in the show notes. And the point is that the computing is expensive. If you can distribute it,
Yeah.
you have all these thousands, millions and millions of PCs that aren't used most of the time.
Correct.
And I like this. I'm going to set my computers to work on this.
Excellent. Plus, you get to see the results, right? I mean, you can watch it happen.
You can see what happens. So they give you a problem, and you can watch it being solved. So in essence, he says one signs up, downloads a client, and the rest is automatic. The client downloads tasks, does the calculations involved, submits the results. Simple for the end user and apparently very valuable for the Folding at Home team and others judging by the amount of publications generated. They have a lot of papers.
Right.
And then he says, my son is hooked on a network game called Platform Racing. in which you collect hats to gain additional powers. The most powerful hats are only given away to those who join the platform racing team in Folding at Home and crunch some 5,000 items. That's a pretty cool and innovative way to gain followers. Why don't you set up a TWiV team and challenge some other podcast? TWiP. We
Like the one that trashes vaccines or something.
Yeah, that was No Agenda.
That's right, No Agenda.
Brain Science Podcast,
How about
Futures
No
in
Good
Biotech,
Agenda?
to beat the TWiV team score. I think we'll do that. I'm going to look into it, and we'll set up a TWiV team.
Make
Maybe
it interesting.
Make it part of the Drobo giveaway. Who knows? We'll see.
Sure.
Anyway, so those are our picks. Sorry, Alan, that got cut out there.
Well, his resonating voice will be back next week, I'm sure.
Got the first 70 minutes of him. He's in there. That's fine.
Absolutely.
We love to get your comments and questions, don't we?
Absolutely.
Keep sending them in, TWiV at twiv.tv. Next week, we'll do an episode of all email.
Wonderful.
Try and catch up.
Wonderful.
You will be here?
Yeah, oh, sure.
Unless you're called to greater business, which I know often happens.
I wouldn't miss it.
TWiV is part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, websites where you can find high-quality science podcasts in a variety of fields and other things and other science resources as well. And do check out our other podcast, TWiP, at microbeworld.org/TWiP. Dickson.
Vincent.
Welcome back.
I'm thrilled to be here.
Racaniello.org.
Yep.
Medicalecology.org.
Yep.
And verticalfarm.com.
Yep.
The media empire of Dickson Despommier. And you didn't know when you started out as a child that it would end up this way.
I had no idea.
Good to have you back, Dick.
Thank you, Vincent.
I like your interactions. They're very astute.
Yep.
And we thank Alan as well.
Absolutely.
Even though he's unreachable at this moment.
Yeah.
Thanks, Alan. You have been listening. Dick, what have we been listening to?
We've been listening to This Week in Virology.
And what is that about?
It's a show, it's a podcast about viruses, the kind that make you sick.
It sure is. Thanks, everybody, for joining and listening. We'll be back next week. Another TWiV is viral.